these are going to be, this is going to be fast, because it's not, you know, you already have a good idea. Basically, microbes are in various environments. We talked about Clostridium in the soil and water. You've got E. coli in your intestines, in animal intestines. You've got microbes anywhere you can think of. There's an example in the book where they have these unbelievably polluted mining pits. And remember, they're always a huge, from ecological or ecology studies, they're always a huge problem. And they think they're so acidic, nothing is ever going to even start living there. And they start looking at it, and microbes are in there, and they can deal with the acidic environment, which is, I think the pH of those things is at times between 0 and 1. So the absolute highest acidity you can imagine. And what it does is affects mainly the factors affect the metabolic enzymes. And another thing they brought up here in this chapter is, or in this section, is a niche, and they call it the totality of adaptation organisms have to make to their habitat. So you have a habitat for any kind of organism, and it means it's able to live in a specific environment, which is its habitat. But it also makes a lot of adaptations, or it adapted to get there. And obviously, the more specialized you are, the advantage is you're not going to have a lot of competition. So if you can live, you know, say as any kind of organism, if you can live in a place where nobody else can live, if you can live in an acidic mining pit that nobody else can touch, there's not going to be predation, there's not going to be a lot of other stuff going on, so it's all yours. But what I like better is, just so you have an idea, because I don't know really, this talks more about how organisms adapt or make adaptations to their habitat, but I would say, or how I always remember it is, the habitat is the organism's address. So where is it? And the niche is its profession. So what does the organism do in there? You know, what does it? What is it taking in? What is it giving out? What is it? How is it interacting with its environment? And a lot of it, you know, that's all metabolism. How does it nurture itself? And what kind of waste products does it produce? And a lot of those waste products are toxins that affect us. So that's when it becomes complicated for us, because those microbes may, you know, be in the soil like Clostridium and live in the soil and the water, but some of the toxins released from the vegetative phase are super toxic to us, or to our neuromuscular synapse or gap. And so we talked about this, that's why we're going to fly through this one. You've got, usually we talk about 3, they call it cardinal temperatures, or you should remember that. There's a minimum temperature, the lowest temperature they can survive and grow. A maximum temperature is the opposite, highest temperature that they can grow and metabolize. And then the optimum temperature is where the fastest rate of growth and metabolism happens. And so in general, it should look, and you drew your own graph, it should look a little bit more like this. And the optimum is not a point, usually it's more like an area where it's hard to say, you know, there's few degrees where they're gonna grow fast and do well. And there's all this area where they're still living, all this where they're still living. And then there could be others with that temperature. There could be others that have the same kind of curve, just in the colder area. Say this is 0 degrees, and you know, this is maybe, if you keep going, this is 80 degrees. So you can have different organisms having their growth their temperature range in different areas, and we'll go through those right now. It's basically what I drew is down here. So you always have an optimum temperature, and there's the three distinctions we already made. Unfortunately, we worked with mesophiles only, so there wasn't a huge difference. And they have their optimum temperature between 20 and 40, and makes sense that most of them are human pathogens because it's the temperature 37 degrees is what we're usually running at, so that's where they have their optimal growth. And then if you're a psychrophile, the optimum temperature is usually under 15, so it would be somewhere here, 15 maybe. And you find those on snow fields, polar ice, deep oceans where the temperature is really low. And the problem is they can grow on refrigerated food, which is usually, that's why we keep it refrigerated, so we have a temperature where food doesn't spoil. And it doesn't spoil because a lot of the microorganisms and a lot of the human pathogens need that kind of temperature. But they're also rarely pathogenic because in general, they're living in environments that are so low in temperature they wouldn't be able to survive in a human body. So they would die before they would have any kind of growth or metabolism in your body. And then we have thermophiles, which are the opposite of the psychrophiles, and the thermophiles it's usually greater than 45. So you can just imagine, if I would think about this, I would say you got 20 to 40, which falls in the range of, not really the range, but it's closer to human body temperature. You go a lot below 20 or 15, you have the psychrophiles, and then the thermophiles are like a little bit above, so 5 degrees up, you'll end up with those. You see them in, for example, volcanoes, compost piles. Does anybody have a compost pile? You stick a thermometer in there, and it's pretty warm in there. It's just from the microbial activity. So from the decomposition, that's where they can thrive. That's a niche. Who else wants to live in a compost pile? So if you're good at that, then you're gonna have a lot of resources. Other ones are other organisms that fall into the thermophile category are the ones that can deal with direct exposure to the sun. And I put this here. It's just a side note. The biotechnology or what they call it, people that researchers in biotechnology are pretty interested in these groups because, for example, Taq polymerase, there's certain techniques they apply in biotechnology that use enzymes that can withstand a high temperature, and they find those in that group of organisms. So they have proteins a lot of times, or enzymes that can survive warm temperatures where every other organism's proteins, enzyme functions would be declining, or the organism wouldn't even survive. The proteins would denature. So that's just why I had that here. You can read a little bit about that in the book. There's also a gas requirement. So oxygen is one of the main gases. We already talked about Clostridium botulinum and that it can live. So it's an anaerobic organism, but a lot of organisms need oxygen. A lot of microorganisms. And what happens is we'll get through these processes, but a lot of times it's utilized and transformed into a lot of different products, and those are usually toxic. So you have, for example, singlet oxygen, you have superoxide ion O2 minus, you have peroxide. We use that as, peroxide was one of the substances we used as a germicide. So there you go, you have a toxic product coming from oxygen utilization. And so what they do is they have enzymes to neutralize those chemicals. So if you are a microorganism that uses oxygen, you have a way to detoxify these products, and that's a lot of times like your superoxide dismutase or catalase, and you can create, and I show you an example, you can create media where you can actually distinguish between aerobic and anaerobic organisms by taking advantage of those differences of what they use. And obviously, if an organism is not able to deal with these toxins, it's forced to live in oxygen-deprived or free habitats. And so these are the different categories. You call them aerobe for utilizing oxygen. Obligate if they cannot grow without it. So if you would have if you sterilize by removing oxygen, that would kill a lot of those, but then you have spores, for example, spore formers. Anyway, that's just obligate means impossible to grow without it. If it's an obligate aerobe, and a facultative means it utilizes oxygen, but it can also grow in its absence. So this is kind of like this is actually an anaerobe. So it should be an aerobe, right? Facultative. Because it utilizes, so I have to fix this. This one uses oxygen but can also grow in its absence, so it should be a facultative aerobe. But maybe they turn that term around. I'll have a look. And if it's microaerophilic, means it requires only a small amount of oxygen. So those are just terms for specific microorganisms and how they use oxygen. And then anaerobe organisms do not utilize oxygen. If they're obligate, then they lack the enzymes to detoxify, and so they can't survive. And if they're aerotolerant anaerobes, they don't usually utilize oxygen, but they can survive and grow in its presence. And so Clostridium botulinum was a obligate anaerobe. So this is one of those cultures that I was talking about. So you have a compound called thioglycolate, and it's you have to remember that all it is if you look at these, it's a reducing medium that can establish a gradient of oxygen. So usually what happens is you have a high concentration of oxygen in the beginning or in the top, and it decreases to the bottom. So in this case you would have I guess an obligate aerobe that grows up here. It cannot live with less oxygen. And then here you have probably a facultative where you have growth with oxygen, but all the way down to here. This one too grows up here on top all the way down to the bottom. So it's also one that grows with oxygen available, but also without. And then you have this one, which you could say is unable to live in oxygen concentrated. Yes.
Back to that other side, maybe it would be anaerobic, because if it can grow in the absence of oxygen, wouldn't that be anaerobic?
Facultative anaerobe.
Yeah, I think that's right.
Yeah. Probably is a facultative anaerobe. I was just wondering what the difference would be of an aerotolerant anaerobe.
Because they don't utilize oxygen. The
Yeah.
facultative can, but
Yeah.
they don't need it.
Okay, so I won't fix it.
So in the first test tube was what?
You would have an obligate aerobe where it cannot grow without oxygen, and it's probably like the gradient, and there in this picture's in the book. The gradient doesn't allow it to grow any further away from the oxygen, so it just grows where the oxygen is highest on top. And then in this case, it's the opposite, anaerobe, where it cannot grow where the oxygen is, and it grows further down where the oxygen concentration is down. And this would be a chamber. This is another one of those chambers where people work, where they grow or they're interested in anaerobic bacteria, and so they work in a chamber that's completely depleted of oxygen. Just as an example. There's also carbon dioxide requirements. Usually there's a, I can't remember the 0.03% of the carbon in the air, but there's some that grow better at higher carbon dioxide concentrations that are normally present in the atmosphere, and you call them capnophile. They really are, and this is like a mini chamber where you might be able to increase the carbon dioxide concentration so that they're fine. But it's just another, there's oxygen, there's carbon dioxide. This is definitely not as important because there aren't that many places where the carbon dioxide would concentrations differ. But oxygen, obviously, you have different, you have the intestines where they're usually anaerobic conditions, or other places, you have Staphylococcus epidermidis on your skin where it's aerobic conditions, so you have different conditions, even on one organism, where different needs are satisfied. And we also talked about, or looked at, we didn't do a pH experiment, but usually the neutrophiles are the majority of them, of the bacteria, and they're living between 6 and 8, a pH of 6 and 8. 7 is neutral, so that's why they're the neurophiles, and acidophiles, alkalinophiles go the opposite way, the extreme pH, like the ones I've talked about in the acidic mining waste pit, and then alkalinophiles, the opposite, where you have a pH above 7 or above 8, and that's where they grow better. And then osmotic pressure, that's, we talked about osmosis for a while. So usually, as I said before, we have hypotonic or isotonic conditions, but then you've got the halophiles, which require a high concentration of salt. So they would be the exception, living in a hypertonic environment. Because hypertonic meaning a high concentration of a solute, which in this case would be salt. And then osmotolerant also makes sense. They don't require a high concentration of solute, but they can tolerate it when it occurs. So they would be likely better survivors in a scenario where you apply salt or sugar to conserve food, because they can actually tolerate it. So we already talked about this as a question I had, but so the conditions when food is preserved with salt and sugar. And what you should also know or remember is when you take a microorganism, even if it lives in the desert or even if it lives in the soil under dry conditions, the conditions can get so dry that they form spores, etc. But most of the time they could be very dry, but the microbe usually has a thin layer of water around it, so there's always exchange going on. And so that's just important to know, because that salt really, that water really has to actually be withdrawn in any kind of situation to kill the microbe, or to make conditions so dire that even that they can't at least reproduce or metabolize anything, so they're just gonna not be efficient.
So what are the conditions that you use for this?
Well, I was mainly thinking about, what are the conditions. I was mostly thinking about the conditions being the environment went from hypo or isotonic to hypertonic, so we have a lot of that, and so it would withdraw water from the environment, and if the environment contains microbes, then it would withdraw water from them, and usually the microbes are at least, they're either dying or their cells would lose water and they wouldn't be able to withstand that at certain concentrations, or at least they wouldn't be in conditions where they can grow, multiply, and then with their toxin affect you. So the conditions are unfavorable for microbes, I guess. There's different ways. I was just putting that in so I remember to ask you that, but a lot of times I already tell you that before I ask you, so that's not good. Other factors, and this is the only one. Barophiles basically are the only ones that can survive under extreme pressure. Most of them live under pressure. You know, in the ocean, there's a little bit more pressure at certain depths. You have atmospheric pressure for most other ones, and then there's some that they found, for example, foraminifera. So it's a protist or protozoa, amoeboid, and it's in deep sea trenches. So there's a lot of pressure. So some of them are able to live in that kind of environment. So those are just a few conditions, you know, pH, temperature, osmotic pressure, and then atmospheric pressure that make a difference or determine the environment for specific organisms. And so these organisms have to deal with these different conditions, and they have different, if they inhabit that niche, they have different ways of doing that. That just gives you an oversight of what they are, and you can tell by just a few examples that they're very successful in adapting to all these different environments, much more so than any other group of organisms or living organisms. Seems like the microorganisms, especially the bacteria and the viruses, are everywhere. I don't think I read that. Oh yeah, so there's one that actually can't see it so well, but there's a microorganism or bacteria that kind of colors snow reddish, so that's what it looks like. So it's all under these? Yeah. Okay. That was the easy one. It just tests, I guess, your knowledge of all the other ones. They have to do with atmospheric pressure, no. Osmosis, no. Microaerophile, I don't know what that is. Alkaline, no. Yeah, okay. Whenever I hear it, I think, okay, it's clear, and then I forget that everybody else, or that some other people might want to know. We'll do a short few slides on those microbial associations, and I think biofilms are part of it, and biofilms are pretty important because they happen everywhere, and they can be a problem if it comes to, say, surgery equipment or I think we talked about hard implants or implants. They could accumulate there, and then when released or if released, lead to a huge problem or disease. And so there's different associations of, and you can imagine it's just like any other ecosystem. Every ecosystem has many players. You have the natural, you have the living organisms, and you have the environment. So any kind of environmental factor. I mean, we're affected by temperature, by pressure, by all kinds of things. We're pretty good at controlling those, but it affects us, and it, where we live, it used to be a lot more what we eat and how we live, and so that's still true for most organisms, and especially for microorganisms. And so there's symbiotic and non-symbiotic relationships. And so if they're free living, And a relationship is not required for survival. They're non-symbiotic, but there can still be synergism and antagonism. And then for those that really require a relationship with each other or with another organism, say in nutritional relationships in general or usually, and they're required by two or more members, then they're symbiotic. And so there's mutualism, commensalism, and parasitism. And most of them you've probably heard about, and we'll just go over them quickly. I mean, mutualism means they both, they live together, they both benefit from it. Commensalism is usually one of them benefits, the other one isn't not really affected, might not even know what's going on. Parasitism is, the parasite is dependent on the host and usually harms the host. If it's a smart parasite, it doesn't kill the host, so it keeps feeding off of it. And then for those two, synergism is members cooperate and share nutrients, and antagonism would be some members are gaining, some are inhibited and destroyed by the action of those that are gaining. It's just maybe you can think of when you try to remember some of those associations, you can come up with an example in the book or and find out. I think there's a few here. So for the symbiotic ones, the ones where you have to have two organisms live together in a close partnership, you have mutualism we talked about. So in this case, for example, you have a marine flatworm, and it has algae, what they call passengers. So this is the flatworm, and really all the green particles you see are algae. And so what it does is it provides, and it's a mutualistic relationship, so it provides shelter, it provides certain compounds for the algae to use in their metabolism. And then the algae, what are algae good at? Why are they green, and what are they good at? They're probably producing energy, or they're producing something by photosynthesis. So they're basically producing oxygen through photosynthesis and some other, glucose, some other organic compound that the worm can use, and the worm in exchange produces, I think, ammonia, and that's what those algae can use again. So there's a relationship where both of them benefit. And apparently that flatworm actually, finds places where there's a lot of sunlight and closer to the beaches, and so it makes sure that the algae are happy, and that's definitely a good example for mutualism. In this case, it's an interesting one. This is like one of those blood augers, and you have two different microorganisms growing on there, and it's an example of where Staphylococcus grows really well on it, and Haemophilus wouldn't grow on it at all. It would not be able to grow an agar, but it can grow really close to this because Staphylococcus uses up the agar and gives off certain products. Sometimes it's toxins, but it metabolizes this agar. As you can see, it's growing really well on this in this stripe. It could be one of your plates where you have just a stripe. So it grows in this area, and then because a lot of the products or the metabolic products that come from Staphylococcus that kind of diffuse out can be picked up by Haemophilus. So that organism would never grow on here, and so Staphylococcus doesn't really care. It doesn't care about Haemophilus, so it's just growing. But this Haemophilus can actually benefit from Staphylococcus. This one is not affected. This one benefits from products of this one. So it's commensalism. And then parasitism. We've seen some. We talked about some parasitic worms. You have other parasites where usually they're using host fluids or just the host, whatever the host provides to to metabolize and to get energy from the host. So those are, and everybody probably seen or heard from parasites. And then non-symbiotic ones, where you have free living organisms that really don't need each other for survival, but they can still be optimizing their life or their lifestyles, and if they cooperate to produce a result that both of them or that none of them could do alone. In this case, they have that would be a synergistic relationship. You have a microorganism, and don't worry about the details here, but it's just a microorganism that can actually degrade cellulose, so it makes cellulose, it converts cellulose or breaks up cellulose, a bigger polysaccharide into these monosaccharides, glucose, and then it gets picked up by a different microorganism because it cannot deal with cellulose. It doesn't have the ability to actually break cellulose down, but it can actually pick up glucose, and then this one produces, as one of the side products, ammonium, and then that can, or ammonia, and that can then be used up by this one. So it's basically those two organisms, if in a synergistic or in a relationship, are able to use each other's products as nutrients. There's probably a lot, or some other good examples that are not.
What about acid free?
You mean when you water them?
No, that we breathe the oxygen.
Yeah. I mean, that.
So you take our
There could probably be a. That would probably go under the other category, because we wouldn't be able to survive. And the question would be, because it's such a long-standing relationship that is probably, you would imagine that, at some point probably oxygen was produced as a byproduct of photosynthesis, and it's just helped evolve any kind of oxygen breathing.
Can those guys survive without the product?
It depends on, but they should be, otherwise they shouldn't be here, because they're not required for survival. So they may have to live, they may otherwise live in areas where they have glucose available, but in areas or in places where cellulose is available, they can live in a synergistic relationship with the cellulose degraders. And antagonism, actions of one organism affect the success of survival of others. So, for example, antibiosis or the production of antibiotics means one organism living in, say, the same community produces antibiotic would be harming or destroying a specific organism. So if they live in a community and you have an antibiotic producer like a fungi, then it would be in that biofilm, it would be difficult for certain bacteria to grow next to it. So what have we, I mentioned a couple. Do you remember, just so we also mentioned a couple, or some of the microorganisms we constantly work with. The one we used for, E. coli is good, exactly. So that would be, microbial flora up here. The one on our skin, probably important. We have, or at least, it could be commensal, where, we don't really care about Staphylococcus epidermidis as much, but they're profiting from living on human skin. Parasitic, so we have different, there's an unbelievable amount of microbes living in and on our body. So all these relationships happen between humans and microbes, and what we're usually interested in is the ones that cause disease, but we're also interested in the ones that we're unable to live without, or that we need for proper digestion, etc. And then to go quickly through or to look at the biofilms one more time. So when they attach to the substrate, and remember glycocalyx, we had a question about that. Sometimes it's like a slime layer. So a lot of the microbes, are able to attach to surfaces with the glycocalyx, and then they basically build up, and then all these different organisms are starting to either accumulate there, and you have different even you have fungi, you have different classes, or everything from starting from orders, or starting from phylums to all the way down to species. You have a huge amount of differentiation, or not differentiation, of different organisms building these biofilms. It's a dominant, or they dominate the structure of most natural environments, and there's some communication, cooperation necessary, and I think that next slide shows that. And they call it quorum, I guess, sensing, or but what it really means, and I don't, you don't need to really know how that actually, or you don't need to know the terminology for this, but it makes sense that you have say a free-swimming organism, and it attaches the way we talked about before, maybe through a slimy surface, maybe through some other way, and then it creates surface for other it creates some other sticky matrix, or has a sticky matrix, and creates the option for other organisms that usually wouldn't be able to attach. And then when they're at a certain density, they're actually able to synergistically to induce through signaling, I guess, to create certain proteins, or to genetically induce the expression of a particular gene, and to then create or to release a certain protein, such as an enzyme. And so food particles can be picked up by this community, because they're in unison, they're producing a protein or an enzyme to digest that. And all you need to really know is, there's lots of different organisms in a biofilm. They're easily built, and they can a lot of times work together to thrive, to get nutrients in, and to thrive. And a lot of it has to do with signaling to organisms to produce a certain enzyme. And then finally, what we've done too is we had a lab experiment, and we looked at growth at different temperatures. We looked at growth at UV light exposure or not. And then what was the final one? Oh yeah, and growth after applying germicides to them. So those are our 3 experiments. And there was a couple of things that were interesting, because here you don't have all the options. So one was, when we look at growth and we try to compare it in our vials, it's pretty hard to say what's minimum growth, or what's little, what's medium. So it shows a little bit of what kind of growth, or what kind of techniques you can use to actually determine growth. And the other thing is, what are the conditions like? I mean, what would you expect if we left those cultures only in there for 12 hours. Is that where they all grow and then they stop? Or did we, you know, if an organism doesn't even grow that much in the first 12 hours, what difference did it make then that to have them at different temperatures? So that's what really, what we're really going to discuss quickly for those, or what we discuss in the next few slides. How does growth in microorganisms usually occur? So there's two different ways. Obviously, the cell can grow, so that grows from grows just like our cells grow, or you have a single cell growth, but you also have an organism growth. So in this case, for microorganisms, especially the unicellular ones, you only have one cell. So the one cell can grow to a certain extent, and then it divides, which is called, and that's important, binary fission. So that's important, and they call it binary fission because it's like basically you have a cell like this, and it's almost like a cut through here where you have the DNA replicated, so you have two chromosomes, and then the rest, all the cytoplasm is divided, so it's a transverse cut, I guess, through the organism. But that's the other way to grow for microorganisms. That's the other. It's the colony. You know, you start off with something you can't see, and a few days later you have a colony. So those are the two ways. You have a parent cell that, and I'll show you how binary fission, or we can look through the next slide. But it basically means you have a parent cell that takes a few hours, sometimes a few days maybe, for a cell to become a parent cell, and then it creates two daughter cells, what they call it, and that's, you go from 1 to 2, and these two do the same thing, so you have 4. Those 4 do the same thing, you have 8. So that's how microorganisms grow, and that happens. We talked about yesterday. Weren't you volunteering, David, to come in every minute and take a picture and see how slowly the colony would grow? So that would be one of them, because it really would happen in I think what they call a generation from a newborn microbe from a daughter cell to being a parent cell. It can be 6 hours. It can be a few days, but it's very little time. So you could literally watch it. There must be something out there where people watch colonies grow before. But what it is you have an early phase of the cycle, so you have a cell, it kind of grows, expands a little bit, and then kind of gets into this stage. It enlarges the wall and the membrane. Remember, you have those two structures in prokaryotic cells, and it duplicates the chromosome. And then there's a inward grow here in the middle of the wall and the membrane, and then you basically have all the rest of the cytoplasmic components, which are not organelles. Usually we don't have any membrane-bound organelles in prokaryotic cells, but the cytoplasm basically divides into these two areas randomly, so you never know what daughter cell gets what, but they both get one of the chromosomes. And then you have two separate chambers, and a lot of times they separate like this, but sometimes they form chains or duplets, so you don't always have them separate like this. So you basically go from one cell through binary fission to two cells. And that's a mechanism you should remember, because it's a very basic multiplication process. Would you think it's a sexual or an asexual? Yeah. Because there aren't genes involved or genetic material from more than one organism. And so it looks more complicated than this, and I'm not going to go too much into this, but you basically have exponential growth, kind of like what the human population is doing right now. But you have you have one organism, or like say you have one CFU. Remember we had those units. What are they called? What was the abbreviation for? We talked about it yesterday. I can't remember the CFUs. Yeah. What was the abbreviation for? Yeah, colony forming. So it wasn't just a single cell, but it was basically. It could be like we've seen here, it could be a cell that's not a single cell, but it could be like say a little chain or a duplet, something like that. And so say you start with one of those, you get 2, and out of those 2 you get 4, 8, 16, 32, etc., etc. So depending on how much time you have and what the generation time is, which can be actually it's on average 30 to 60 minutes, so that's how fast it goes from a daughter cell to reproducing to another one, so that's how fast they produce. So it goes, it depends on the generation time, because it can be between minutes and days, and it depends on how good the conditions are, but basically the generation or doubling time can be calculated, and usually it's you start off in the beginning. The growth is obviously it takes longer because it's exponential. You have an increase, an exponential increase in cell numbers, and we can, if you want to know how to calculate that. So if anybody says, well, I don't know what I know I took a colony, not a colony, like I said, you would, I know we started off with, we're separating a certain amount of numbers of cells. So you would have a Ni, which is the initial number of cells, and I think there's an example next we can go through, and then, and you want to know the total number of cells in the population at a specific time point. And you know the generation time, so you know, oh, these guys had 3 hours to grow, and they build new generations every 30 minutes. After 3 hours, you already have 6 generations. So if you know you start with 1, you know there's going to be 2, 4, 8, that way basically. You can calculate that by this formula. So if you go to the next one and you say, E. coli has a doubling time of 20 minutes, so the generation time is 20 minutes. And there you have 5 cells to begin with. So you would put that into the equation. You start with 5 in here, and 2 to the 3 hours. How many generations is that? 9 generations. So if you go back, 2 to the 9 times 5, because you start out with 5. What is, I don't have a calculator. Anybody have a calculator to find out how many cells we have after 3 hours? Times 2 times 2 times 2 times 2. Don't plug it in that way.
We did the 9 is 512, and then times 5 is 2560.
Okay, thank you. 2560. So that's the number of cells you would have after 3 hours of E. coli. And you started off with a number that you were unable to almost, I mean, if you remember your plates, this is nothing. And this is how fast you get them after 3 hours. And you get millions quickly, and that's how you actually get them visible. When you look at the growth that you had in your plates, a lot of times you work with bacteria that are invisible, and after a few days you have these good-sized cultures, and that's what that is. So you can go the opposite way too and say, how many did I start out with by looking at, basically counting cells and saying, oh, I got 500,000 cells in this culture. Yes. 9 came from 9 generations. So if you know that one generation takes 20 minutes, then you have 3 generations in an hour, and 3 hours later, they had time for 9 generations. Does that make sense?
Yes.
It's just emphasizing how, if you think of generation times of 20 minutes, it tells you how quickly they're able to grow and reproduce. And the problem is a lot of times you don't know what you start with, and you might know the generation time, but even that is not very clear because the conditions aren't always optimal, and really it depends a lot on. If you ask a technician, they would try to get you really good growth. They ask, you ask for E. coli or something, and they say, well, this one didn't grow very well, or the culture was too old, or so you don't really know if the generation time really 20 minutes or is it delayed because the conditions aren't great or whatever. So what you could do is you can actually do a plate count, and this looks complicated, and there's one mistake in the book that I think it is a mistake. I'm always questioning myself, but basically what you do is you have a flask inoculated, and it's say 500 milliliters, and you want to know what the growth is, and so after 60 minutes you take a small amount, you take 0.1 milliliters, so 5,000, I guess. You have 500 milliliters total in there. So if you take 0.1 milliliters, that's divided by 5,000. And then you look at it and you put it on a plate, and then you get basically 1 colony grows, and then you do that again after 120 minutes, 2 colonies grow. Then you can basically say, okay, if there were 2 in there, and there were 4 in there, and 7 in there, and after 240 minutes there were 13 in there, you can calculate back and say, well, if there's, for example, and that's how I think why this is mistaken, if there's 2 in there after 120 minutes, then that would be 2 in 0.1 milliliters equals 10,000 in 500 milliliters. So in the 500 milliliters you would have 10,000, and it should be like that. It's just a way of it's just a technique. So if you find 2 in 0.1 milliliters, that would mean you have 10,000 because it's multiplied by.
Figure out the ratio.
Yeah, it's just a ratio. And it's one way to do it. It's also still very complicated, and doing that really well, getting really good results on the plates. I don't know how much so you basically estimate the total cell population in this flask. But what usually happens is, and I said that before, if you have what usually happens is it's hard to know how well the colony grows, and so what usually happens is you have a growth curve, and it starts with a lag phase where the cells are adjusting, where they're enlarging. They're not actually doing binary fission already. They're basically just getting bigger, and then eventually they get into this exponential growth phase. So now you have all the conditions right, and they start replicating, they start multiplying really fast. But what do you think happens up here? Like, what do you think happens? Why wouldn't that go on forever? Why are we worried about the human population growth being exponential, and what do we think happens with us? Yeah, so we run out of resources. We either produce too much waste, that's going to go somewhere. We don't have the resources, so there's a point where it goes into a stationary phase, and then could go into a death phase, depending on how you deal with it. And that's the same for any organism. I mean, that's why we're worried, because we know that from everything else. So you have a lag phase where there's little growth. There's the cells are getting adjusted to the new environment, and they're pretty healthy. So in a good culture, and then you go into a exponential growth phase, so maximum growth. So you need adequate nutrients and favorable conditions. And I think in some cultures what they do is there's a constant flow of nutrients in the laboratory and a constant cleaning of the culture to get rid of the waste, and that's how you keep it biotechnology, how you keep microbes producing for you all the time. So they keep it basically a constant, they stay in here by constantly keeping the substrates coming, or the nutrients coming and the waste going out. And that's ideal, you know. That's how we would go forever. If somebody would constantly supply and take our waste away, we could just, you know, I mean, reproduction is not a good example for us, but we could just, it would be very simple. But no system, no ecosystem ever does that. It's fine in a laboratory setting, but not in a natural environment or an ecosystem. So at some point, growth or new organisms and death of cells are equal, because nutrients are depleted, and then you have pollutants, and then you have a lot of other compounds that are building up. And then the death phase, usually cells start dying exponentially because the limitation increases and the pollutants are building up. But this is usually not as steep. This is usually not as steep as the exponential growth phase. So this is where usually you end up with this, and then it could, depending on the culture, it may be completely going to zero, or they might recover. And there's two more things, and then we're done with this. Where basically, if you want to look at that, which is what we did here, you look at turbidity, and what we should have is one of those readers where you take a cuvette or a tube and you stick it in there, and you just look at how turbid they are. Basically, what we did with our eyesight, looks clear, looks turbid, looks really turbid, is what a machine measures, and so it reflects the population size, I guess, or the amount of microorganisms in the culture. It's clear that you know, it's a pretty simple concept. And then there's two other ways. You can enumerate bacteria by looking at viable colonies. So a lot of times when you count them, the problem is you don't know which ones are alive and which are dead. So they've gotten really good at automating it, and there's automation, or there are automated systems where you basically the sample is in liquid, and it falls through this or goes into this tube and going through a certain area, and there's a detector that can actually count the cells for you, and it also can differentiate. So it can say, that was actually a red cell, that was a white cell, that was a dead cell. So they're going really far. But the counter, I think we're going to manually count in one like this. And I haven't tried this one yet, but we'll get to this one. Just so you know this is what that's for. So we'll be able to count cells through those. And then we can get into detail when we do that exercise. So I'll give you 9 minutes. Is it a lot today? I'm not trying to make this harder than other times. I think it's harder. You know, I was gonna say today is 4 weeks. You've gone through. We've gone through half of it. Yeah. That's really big for me.